All right, sorry, I had to grab a quick drink there. So our next section here is on chemical reactions and enzymes. And these are very important because basically our body and our cells continuously undergo chemical reactions. And a lot of these chemical reactions involve things called enzymes. So let's do a quick review here of what exactly chemical reactions are. Chemical reactions are made up of two components. First of all, we have the starting items that we work with, the reactants. And then we have this arrow, which indicates that they're undergoing a chemical change. Basically, the reactants kind of get pulled apart, at least partially, and then they kind of get rearranged and put back together to make the products. So for example here, I have our reactants, our carbon dioxide and water, and so they get pulled apart and rearranged to make H2CO3, which is carbonic acid. So the way that you would read this is CO2 plus H2O yields H2CO3. And the yields indicates that we have a change happening. So again, our reactants are the carbon dioxide and water, and our products are the H2CO3. And this is a very common reaction that takes place. When your cells release carbon dioxide into the bloodstream, this is what happens to it. Now, in order for these chemical reactions to take place, we also have to talk about energy. There are two types of chemical reactions. There's either the ones that release energy or the ones that absorb energy. And these energy forms can be heat, which is the most common one. There can even be sound or even light. If you think of like a lightning bug, they have some chemical reactions that go on that allow them to flash and generate light like that. Now, because we need energy for some of these reactions, that's why we have to have a source of energy in terms of our food. And when we refer to all of these reactions that involve the energy that we bring into our bodies, those are our metabolism. So, whether we absorb energy or release energy in a chemical reaction, all types of reactions, both sets of them, have to have a little bit of energy to get them started, because they don't occur spontaneously. So I can't just put these things magically together and have them react. So the energy needed to get a reaction started is called the activation energy. And the way I think of this is, you have a ball sitting at the top of a hill. You know that the ball is going to roll down the hill, but if it's just sitting there at the top of the hill, it's never going to roll down the hill. It can sit there forever. So you have to give it a little shove, or you have to give it a little push, and that little push that you give to it, it doesn't have to be big, but that's its activation energy. You're actually adding a little bit of energy to the ball to get it started so that it rolls down the hill. So we can see here on these diagrams, we have one that absorbs energy and one that releases energy, and both of them have this activation energy that is a part of it. So if you move over to your handouts packet, excuse me. So the graphs below show the amount of energy present during two chemical reactions. The top graph, which is the one that I have on the left, has been labeled to show the reactants, the products, and the activation energy. Recall that activation energy is the energy needed to start a chemical reaction. So we need to, on the other graph, which is the one on the right, on your worksheet it's the one on the bottom, we need to label the reactants and products on the bottom graph and then draw the arrow to show the activation energy. So our reactants are here because that's where we start, and our products are here because those are at the end. If we look at the one on the left, the activation energy goes between this bottom line that corresponds with the reactants and then the upper line at the top of the hill. So here, our bottom line is actually with the product. So our activation energy actually goes from the reactants to the top of the hill. So that is our activation energy. I abbreviated just because I'm writing on the board and there's not a lot of room. You have easier time writing smaller on your paper. All right, so down below, the question. Which type of reaction often occurs spontaneously? Those are the energy releasing reactions that you would want to circle there. So, now that we kind of understand about activation energy and chemical reactions, let's bring in this concept of enzymes. Enzymes are what we call protein catalysts. And what does a catalyst do? It is something that speeds up a reaction without getting used up itself. So it makes the reaction go faster and it's reusable. So where specifically enzymes speed up our reactions is inside of our cells. So how are they able to do this? Well, what enzymes do is they lower the amount of activation energy that is needed for a chemical reaction to take place. So if you look in your notes, you have, sorry, I'm just checking here, kind of a blank space there where it says sketch of a reaction pathway. So you want to kind of sketch out something like this. Down here at the bottom where it says course of reaction, you can actually just put time. Now, you may want to pause this so you can sketch this out, and then we're going to be labeling some things, as you can see. All right, so the top line, the beginning part here, just like before, this is the reactants, and the ending point here, that is the energy of the products. Now, the top line is the reaction pathway without the enzyme. So if you want to think of it, let's actually do a physical correlation here. So you could walk from the reactants all the way up and over the top of the hill and then walk down to the other side to the products. So the activation energy that we would need is this one, this bigger one. It takes a certain amount of energy for us to get up to the top of the hill, and then walking down the hill is really, really easy. But there's also another option with this, and that is to use an enzyme. And an enzyme is that energy shortcut. It lowers the amount of activation energy, and that's represented by the bottom line. So again, physical relationship. So let's say we're going to walk that same path, but instead of going all the way up and over the hill, there's a tunnel about halfway up. That makes life a lot easier. So I come out the other side and I go down, so I end up at the exact same place, but I get there faster because I don't have to go all the way up the top of the hill. That's because I'm using less energy too. So the activation energy without the enzyme is less. Now, enzymes are what we call specific. They only catalyze one particular type of reaction. They don't necessarily do all types. They're not a one-size-fits-all type t-shirt. They can only do specifically what they are intended to do. And enzymes are reused. They are reusable. So the cell can use them over and over and over again. Eventually they do wear out because they are biological molecules, but they can be used quite a bit before that happens. So let's talk a little bit about this specificity. And there's a new word we want to talk about, and that is substrate. In order to talk about this and how they are specific, we're going to talk about what we call the lock and key model, but we're going to use this new term, substrate. Substrate is just a synonym, another word, for the reactants in an enzyme-catalyzed reaction. So anytime you see the word substrate, you're going to need to know that there is an enzyme involved. We don't use the term substrate otherwise. So how does this lock and key model work of specificity? Okay, an enzyme shape fits the substrate's shape, and it works just like a lock and key. So, on my keys here for school, I have a key that opens my door, it opens the bio prep room, it opens the chemical storage room, it opens any of the science doors that we have. But if I were to try to go and unlock, let's say the library, my key won't work there. It doesn't work in the office. It doesn't work in Mrs. Shirk's room or any of the math classrooms. It's specific to the locks here in the science department. It won't start my car. It won't open my house. So the shape and the role of an enzyme is actually determined by the arrangement of the amino acids. Enzymes are proteins, remember. So the amino acids and how they fold on each other determine the shape of the enzyme. Now this folding creates something that we call an active site, and this is where the enzyme and substrate come together and they meet during the reaction. So let's kind of sketch this out. All right, so I have a substrate here. This is my reactant, and I have an enzyme. And if you look, notice how the shape of the enzyme should fit here with the shape of the substrate. That is really, really important. If those don't match up, this whole thing stops. It doesn't even work. Okay, So in this case though, the shapes do match up, so the enzyme and substrate come together, and you can see they meet up and they fit together like puzzle pieces to form the enzyme substrate complex. And this exists while the chemical reaction is going on. Where they meet is the active site. So that joining line here, where they are actually kind of touching, is that active site. Now, once the reaction is over, the products are released and the enzyme separates. And it can go back and get reused. Okay, so on your handouts packet, you have a worksheet that deals with enzymes. Many chemical reactions in cells take place on enzymes. These reactants are called the reactants bind to the enzymes until the reaction is complete. These reactants are called substrates. When the reaction is complete, the products are released. So, we have the two substrates here, and this is our enzyme and the active site. You can see the substrates come in and they join in here on the active site. There's the chemical reaction that takes place. We get products that are released, and then the enzyme can go back and everything gets reused again. So let's place these three things in order. Okay, so the first step is that substrates have to bind to the enzyme if we look at the diagram. And then the second step is that they get converted into products, and then the products get released. All right, so where do the reactants bind to the enzyme? If you look on the diagram, that happens at the active site. Now, what is the function of enzymes in living things? They are catalyzing or speeding up chemical reactions. Now, back over in your notes, there's a couple things we need to keep in mind with regards to enzyme action. Enzymes can be affected by conditions inside of the body. The two main ones are temperature and pH. What you have to understand is that there is a best temperature, an optimal temperature, and an optimal pH for enzymes to work in. For most enzymes, this is 37 degrees Celsius, which is our body temperature of 98.6, and a pH of 7, which is about neutral. Now, there are exceptions. Just depends on where you are in the body. If we are above optimal temperatures, the active site denatures or deforms. Same thing if the pH is off. If it's too acidic or too basic, the active site will also denature or deform. So let's go back to my school key here that opens up all the science rooms. If it gets too hot, now it'd have to be pretty hot, but if it gets really, really hot, I could actually melt that key. Now I haven't changed the fact that I have the same amount of metal that made up my key, just like I haven't really changed the stuff that has made up my enzyme. I haven't broken it in any way, I've just melted it. It's not going to work anymore in my door or any door that it normally would open. And that's exactly what happens to the enzymes, is it stops working. So the chemical reactions either go very slowly or they don't go at all. Now, what you can see here is that with pH and temperature, we have this peak, and this peak is the best one. So if you see a graph like this, you want to pick the peak. If they're asking for the optimal, you want the best. It doesn't mean that it doesn't work with a lower pH or a lower temperature or a higher pH or a higher temperature. It just doesn't work as well. Denaturing is a very common question on the keystone. Now, there are a couple other factors that also affect it. That is the amount or the concentration of either the substrate or the enzyme. As the concentration increases, so does the enzyme activity, but at a certain point it will level off, kind of like on this graph. So that is the end here of our discussion of enzymes and chemical reactions.